My name is Graham Raby. I'm an assistant professor of biology at Trent University in Canada. I'm also associate editor at Conservation Physiology, and so is our speaker today. So welcome to today's Conservation Physiology webinar. Our speaker is Dr. Anne Todgham, who's going to be telling us about her work on developmental effects of temperature and hypoxia in Chinook salmon. Anne has a really impressive CV and has done a lot of interesting and diverse work in animal physiology. She's currently the Department of Animal Science at at UC Davis, and is also the chair of that department. Anne did her bachelor's at Guelph University. Like many good fish physiologists, she originated from Canada. And she did her PhD at UBC and then did her NSERC postdoc at UC Santa Barbara before starting as a faculty member at San Francisco State University in 2009 before moving to UC Davis in 2013. Anne is an environmental physiologist who works on sort of figuring out how animals cope with environmental change, with a focus mostly on fishes and shellfishes. And a lot of the work that her team does is relevant for threatened species or populations, relevant to global change biology, or for sustainable aquaculture. So I'm really looking forward to hearing about your work on Chinook salmon today, Anne, and please take it away.
Great. Thanks very much, Graham. And I'm just, and welcome everybody. Thanks for tuning in. And yeah, it's exciting to be able to present some of the work that we published in Conservation Physiology a couple years ago. So today I'm going to tell you about the differential sensitivity to warming and hypoxia during development and the long-term effects of developmental exposure on early life stages of Chinook salmon. And of course, I'm giving the talk, but right, it's rarely just a single person show. This is the work of my past PhD student, Annalise Del Rio, and that was paid for by the Delta Stewardship Council. So she received a nice graduate fellowship that pays both your stipend and for your research. So it's a really nice program that we have in the US, in California. And what else I think is great about this project is that Ben Martin and Rachel Johnson both were part of the National Marine Fisheries Service, and Josh Israel is at the US Bureau of Reclamation. And so it represents a really nice collaborative effort between sort of university scientists and agency scientists. And it was such a great sort of collaboration for Annalise that she now actually works at the Puget Sound salmon program as an environmental scientist in conservation. So as Graham kind of outlined, my group is really interested in understanding sort of really broadly from a physiological perspective, how good are animals at multitasking? And we take sort of a number of different study organisms to be able to understand sort of unifying themes in this question. Right, and so to give you a sense of the work that we do, it's predominantly in fishes, but we also have work in shellfish. So this work is focused on species that are not only of conservation concern, like basically our delta and our longfin smelt in the San Francisco Bay estuary, our Chinook salmon, our rockfishes on the coast, but also has applications for aquaculture in terms of white sturgeon production. These, of course, are California mussels, but we do work on some of the aquaculture mussels, and then both work on native oysters as well as Pacific oysters for aquaculture. And I don't need to sort of spend too much time on this, but right, as conservation physiologists, I think we sit at this really nice nexus between physiology and ecology, because what we're really interested in understanding is how animals integrate environmental signals and how changes in environmental conditions impact the physiology of the organism from a really integrative perspective, starting at the genome all the way up to the whole organism, and how that can result in changes in performance, which might be changes in growth, metabolism, stress tolerance, can be behavior, which ultimately impact these sort of ecological processes at both the population and community scales. And what we're, as a team, really beginning to understand is that changing only one aspect of the environment does not tell the whole story. So that if we want to take, if we want to use the science that we're generating in our labs to provide information to managers about vulnerability to environmental change, we need to be thinking about multiple stressors, because animals live in very dynamic environments, and it's not just temperature or not just hypoxia as examples that are changing independently of one another. Oops, sorry. So this multiple stressor perspective is really essential. And I hope to sort of highlight this with this really sort of basic set of figures here that, when you take stressor A that has some impact on performance and stressor B that has some impact on performance, and when you add them together, you can get a diversity of responses, right? So it can be additive, that basically 1 + 1 is equal to 2. It can be antagonistic, where the effect is less than what would be predicted when you would add them together, right? So in this case, 1 + 1 equals something like less than 1. Or it can be synergistic, when the effect of the stressors in combination is much greater than those stressors when simply just thought about as being added together. And what we know from ecological literature and an accumulating body of literature in the physiological realm is that about 75% of multiple stressor experiments demonstrate these non-linear interactions between stressors. And so as a physiologist, that's fascinating to think about, like, why? Well, how do stressors that, when exposed independently, have one effect, but then combine together through mechanisms to either cause antagonistic or synergistic effects? But on a sort of more basic level, what this tells us is that we need to be careful about doing single stressor experiments and then just adding the results together of those two experiments independently and saying, okay, this is how the animal is going to respond if you put those two stressors together. So our predictive power is strengthened by these multiple stressor experiments when they're appropriate for the species. So a lot of what our group has been doing is really trying to use a number of different study systems to really highlight the importance of incorporating environmental complexity into our conservation physiology research. And this is because in many times our experiments ask what animals can do rather than what they do if we don't take some of the natural environmental variability and dynamics into our experiments. We all have a very important role as physiologists at the table for providing sound science for management and policy. And as I've been saying, organisms live in these dynamic, multivariate environments. And so we need to have this sort of, in our experiments, this balance between ecological relevance, robust science, and the comfort level with uncertainty, right? So a lot of times people say to me, well, why don't you just do all your work in the field? Well, it's really complicated. So going back to the lab allows you to start looking at more discrete effects. And so, right, stressors interact in these nonlinear ways. And so by incorporating not incorporating environmental complexity, we could have the tendency to underestimate or even overestimate the vulnerability of stressors if we don't incorporate ecologically relevant features of their environment. And by being more ecologically relevant, then we can provide more actionable management strategies to preserve organismal function. So many of us in conservation physiology take a bioenergetic approach to understanding the impacts of stress. And I like to sort of use Ina Sokolova's kind of diagram to understand how to think about balancing energy supply and demand, and how stressors impact energy that can be allocated to these different sort of biological processes, right? So in this blue down here, we have basal maintenance, and that's basically what it costs to keep the lights on in an animal, right? Keeping ion gradients, circulating blood through your system, breathing, all of that has some sort of basal cost, and that's this maintenance cost. And then we have all these other functions like activity, growth, reproduction, or development, depending on the animal's life stage, and storage, that are these other aerobic scope functions that sort of, as physiologists, we use as proxies for fitness, because they give us some insight about how a stress might impact fitness. And for an animal to be successful, they want to balance energy supply and demand, because when demand outpaces supply, that's when things start to go awry. So when we think about, so that was unstressed conditions. Under stress conditions, there is this increase in basal maintenance costs that are used to deal with sort of fund this, I say fund, provide energy for the stress response. And there are increased costs associated with circulating blood and pumping blood. And what that does is it shrinks the amount of energy that's available for these really important aerobic scope functions. And in some cases, depending on the type of stressor or depending on what's happening and the level of the stress, we can have a reduction in ATP supply, further sort of truncating the amount of energy that an animal can allocate to these important aerobic scope functions. In our group, we spend the majority of our time looking at early life stage physiological performance, because we know that sensitivity to environmental change can vary across developmental stage. So this is an example right here of a flatfish where you take the CT min and the CT max, and you look at this sort of this tolerable thermal habitat across the full range of animals, which is incorporating everything here, the highest high and the lowest low. But if you break it down from embryos all the way to adults, you can see that there are these discrete differences in tolerance ranges. And these early life stages tend to be the most vulnerable to stress, or the most sensitive to changes in environmental conditions, temperature in this case. And that's because they have poorly developed defenses. Their cardiovascular system is still developing. They have a lot of energy that needs to go into growth and development, and less energy can be allocated into defense mechanisms. So if you're interested in understanding kind of how do animals respond to stressors differently across life stages, Salmonids are a really good study system, right? So we're really familiar with these sort of anadromous fish where adults return from the ocean, they swim upstream into our freshwater river systems, they spawn eggs, which then hatch into these alevins, eventually become fry. These fry begin to outmigrate back towards the ocean, undergo a process known as smoltification to become sea ready, and then they spend basically between sort of 2 to 3 years, depending on the species in the ocean, before the whole cycle is repeated. And so if we think about Chinook salmon specifically, in California, they're at their southernmost range, and their populations are declining due to dams, climate change, drought, habitat loss. And we have many management efforts and actually funding that's targeted at salmon populations. But what's important to understand, and I think this is true, all over the range of salmonids, is that there is this delicate balance between competing water demands, right? So as conservation managers, they want to maintain as much water in the river system as possible to support the diversity of fishes and invertebrates that live there. But in California, we have, I think it's at least 10 of our most important crops like lettuce and tomatoes and come from California. And so we need a lot of water for our agriculture. And then we have these huge city centers like San Francisco, LA, that also are competing for water. So in California, we have 4 runs of salmon, of Chinook salmon, and their names are based on when they migrate into the river for spawning. So we have the winter run, the spring run, the fall run, and the late fall run. The winter run is our endangered run. The spring run is our threatened run. But the fall and late fall run are currently our most abundant. And so the majority of the work that we do on salmon in California is on fall run and late fall run, although we're lucky to be able to, because we work with conservation agencies and management agencies, we are provided access to winter run and spring run salmon for work as well. And what I want to show you here on the right is sort of the spawning habitat for our Central Valley, which is the part of California that I live in, right? That's this central part right here. So even though California is, we mostly see what's happening in the coast in San Francisco or down in LA, right? We have this really hot desert environment that sits in the middle of California. They're called the Central Valley. This is basically the where our fall run and late fall run salmon are able to spawn. This orange region is the historic spawning range, and the green region is our current spawning range. And that's because we've basically dammed pretty much every river in California, and this restricts these historic cold water spawning range sites from our salmon, which is putting them under increased threat because these other river systems are becoming quite warm due to both drought and climate change. So I want to bring you back to these early life stages, and our work is focused on these early life stages here, from embryos to fry. And for those of you that aren't as familiar with salmon, eggs are fertilized in these gravel reds, which are basically these gravel nests. And they are buried there for about 2 to 6 months. And so, as you can imagine, right, they're embryos, so they have pretty much zero mobility, although they do, when they get to a certain stage, start spinning in their chorion to help kind of circulate the fluid. And they have finite resources in their yolk sac, right? They're not feeding. The conditions in those reds are dependent on gravel size, sediment input, interstitial water flow, groundwater inputs, and can be quite different from the rivers that's just right above them. And these gravel reds tend to be low flow, low oxygen environments, and with spawning sites now restricted from the cold water reaches, are warming at quite a rate. So that sort of formed the foundation of Annalise's PhD work, where she was really interested in understanding the impacts of warming and hypoxia on early development in Chinook salmon, because these early life stages are known to be thermally sensitive and prone to high mortality. At the time of her work, management efforts for salmon embryos were really focused on water temperature. But what we found is if you took the mortality data and the performance data from temperature experiments, they didn't do a great job at predicting hatching success and outmigration success in the real world. So we sort of thought, well, maybe hypoxia is also involved in this equation and could be causing embryo mortality that's not accounted for by temperature alone. So I'll bring you back to this Sokolova work to remind everyone that what warming does is warming really raises the floor. So what it does is it increases the basal metabolic costs, restricting the sort of aerobic metabolism that can be used for these aerobic scope functions. And I'm particularly interested for this study in growth and development. But that hypoxia further kind of truncates this, this available energy because it reduces aerobic it reduces energy supply. So looking at warming and hypoxia is a very interesting multiple stressor or double stressor scenario because it acts in different ways on bioenergetics. So this is work that we did on late fall run Chinook salmon, right? So an abundant group of Salmonids. We got embryos from the U.S. Fish and Wildlife Service, Coleman National Fish Hatchery. We got about 2,000 embryos from 6 different families, and we reared them in replicated tanks that sort of look like this, where we can keep individual embryos for the most part separated, although sometimes they buddy up. And we raised them under sort of a combination of 2 different stressors, so 4 different treatments, where we had both 10 degrees Celsius as a control temperature and 14 degrees Celsius as a warm temperature, and then normoxia in terms of 100% saturation, and then hypoxia with 50% saturation of air. And this was Annelise's first paper that she published in Conservation Physiology. And I'm just going to show you what happens to hatching success, in part because I want to get to our more recent work, but also in part because this figure alone changed how managers think about oxygen in their environment and are now trying to think about more than just temperature. So it really was an important piece to conveying the importance of hypoxia when considering early development in salmonids in our river systems. And so what I'm showing you is percentage hatched on, so it's really basic, right? How many hatched in the different treatments. This is our normoxia treatment, control temperature and warm temperature, and then our hypoxia treatment, control temperature and warm temperature. And you can see overall that hypoxia significantly reduced hatching success, and that this was amplified if you were under warm conditions, to the point where we had basically about a 10% hatching success rate. And that's more in line with what we're seeing in the wild. Right, so I'm going to bring you back to this slide and remind you, well, I mean, maybe there are things that we can do to manage water releases such that we can, right, so we have this delicate balance, right, between water for conservation versus water for agriculture. And maybe we can release water at certain times of the year when there's critical windows of sensitivity for salmonids. And so to be able to start to get at the idea of, do targeted water releases have the potential to mitigate the impacts of red conditions on early development in salmonids, we needed to sort of go back to the lab and say, are there different periods of time that during that embryonic period where embryos have different sensitivities to warming and hypoxia? So this is the paper that is more recently out of Conservation Physiology in 2021. And so what we did was, instead of just having we had the same experimental setup we had it before, but instead of having chronic hypoxia or just chronic hypoxia, where basically from fertilization all the way to hatch, they were exposed to that condition. We took certain periods, so 5 days after they reached the eyed stage and 5 days after they reached the silver-eyed stage, to see whether or not there were critical windows of sensitivity within this embryonic period. And so we did that for all the different treatment combinations. This was, I guess, my students, they say I'm notorious for working with them to come up with the most complicated design. But Annelise was a champ, along with Gabby, the undergraduate that helped, and they were able to execute this experiment really, really well. So we have all combinations of early hypoxia, early warming, early warm and hypoxia, late, and then chronic. And these chronic are very similar to what we had in the last experiment. And then we were interested to know whether or not, right, because once they hatch, they return into the river system, and so conditions are going to be more stable. We wanted to know whether or not there were any carryover or persistent effects from these early rearing conditions if they were returned to ambient control conditions and monitored at the alvin and the fry stage. So this is a similar diagram to what I showed you before, where it's percentage hatched in what would have been before the hypoxia alone, warming alone, and then warming and hypoxia together. What's interesting, this is done in a different year. These are runs that were 2 years apart. And so we don't see this drastic decline in the multi-stressor group like we did before, but we see a lot more variability depending on the family. If we add the other groups in, what's interesting is that the greatest sensitivity to multiple stressors seems to be just before hatching in this late warm hypoxia group. That's where you have the lowest percentage hatched. We were interested in knowing a little bit more about how this affected, right, the rate of development not just percentage survival in terms of hatch. And so we looked at how many days post-fertilization it took to get to the eyed stage, the silver-eyed stage, and then how long it took for them to hatch. And again, right, so hypoxia is in these three, warming alone is these three, and warming and hypoxia is these three down here. And what I hope you can see is that this chronic warming and hypoxia led to significantly faster development times, which is not overly sort of, yeah. So anyways, it's something, it's something that we would have predicted. What I think was maybe what we wouldn't have predicted is that if you look at how much time it takes to get to the fry stage, and so in this case, this is the button-up stage for us. So when they've fully absorbed their yolk sac, you can see that, there's still an effect of chronic warming. That chronic warming still gets to the fry stage the earliest. But if you look at how long it took 39 days from hatch to fry stage. And in the control, it takes about 34 days. So when warming is removed, the rate of development sort of stabilizes, and even, it's actually even somewhat slow than you would predict. On the other hand, hypoxia, if you look at chronic hypoxia, it actually significantly delays the time it takes to get to the fry stage. And again, right, they hatched at about on average, about 53 days post-fertilization, and then 41 days later reach the fry stage. If we look at growth metrics, you can see this is percentage of body tissues at 5 days post-hatch. So this is alvin. Basically, this is the relationship between body tissue to yolk. Basically, how much of there is still yolk versus how much of what is there is the body tissue. And so, I've just taken the data from the figures before and popped them on here. But what you can see is that chronic hypoxia, which is this one here and this one here. So chronic hypoxia alone, chronic hypoxia and warming results in smaller alvins. So this is a persistent effect of chronic hypoxia on sort of growth rate after they're removed and put back into ambient conditions, but really only for about 5 days. If you carry this out to fry, right? So this is now kind of 73 to 94 days post-fertilization. There's really very little effect on condition factor, right? So the condition of these fry is very similar. I will note though that these chronic hypoxia individuals are still smaller, but from a condition perspective, they're quite similar. Now, what is somewhat surprising, although we've seen this before, is that there's actually some benefits to being reared under these sort of, I guess, more poor conditions. So rearing in these early rearing in warm conditions, so this is all of the warming groups, resulted in these persistent increases in stress tolerance, right? So I'm looking at early warm, late warm, and chronic warm, these orange colored ones. They had an increased thermal tolerance, so this is CT max, higher than control if they were raised for different amounts of time under these warm conditions. There's also, you can also see this in the chronic warm hypoxia group too, right? So this warming increased thermal tolerance, not only in just the only thermal tolerance, but also the thermal tolerance in warming, but only in the chronic exposure. And our hypoxia tolerance is much more variable because this is time to loss of equilibrium. So the greater the time, the greater the hypoxia tolerance. But what you'll see is that there's also this sort of increase in hypoxia tolerance in this warm group as well, which is significant. Well, it's not significant, but which is a little bit tighter in the chronic warm group. So to summarize, right, so sensitivity to multiple stressors seems to be greatest right before hatch. And this isn't surprising because the greatest embryonic metabolic demands are right before hatch. They're really starting to grow. They're really moving in their chorion, and they now are starting to, right before hatch, they're starting to get the muscle mass to be able to hatch out of their chorion. We noted that chronic warming sped up embryonic development, but those effects were not persistent on development once you removed them from that warm environment. So it tells us that it's probably the result of thermal kinetics or dynamics on reaction rates. So like, growth and development, rather than more permanent mechanisms of action, is how temperature is working in terms of development and growth. And this is a little bit different than chronic hypoxia, because chronic hypoxia had little effect on embryonic development, but then the effects, these carryover effects, emerged after they were removed from the environment, and you looked at rates of development and growth in Alvin and Fry. So that's suggestive of a more permanent mechanism on biological processes. I don't know what that is, but I would harbor a guess that there's some of these really interesting epigenetic mechanisms at play here. And then last but not least, chronic warming during early development led to more stress-resistant fry, these carryover effects. So there is some benefit to having these early kind of more poor water quality conditions early in life. And that chronic warming, while it doesn't seem to have these sort of permanent mechanistic effects on development and growth, that there may be a more permanent mechanism on stress tolerance, cellular pathways, or there's a decreased sensitivity to stress. And how long this lasts, we only took them out to about anywhere from sort of 73 to 94 days post-fertilization. So I don't know how long that lasts. So in conclusion, I hope that we've shown, and we've been, the fact that we work with managers on this, that dissolved oxygen should be considered in management plans in addition to temperature, and that when we think about managing flow, we're not just thinking about managing temperature, we're also thinking about managing oxygen conditions. We've shown that there are critical windows of sensitivity, so that might allow for more targeted releases, but given that these river systems have 4 runs of salmon, it's actually more complicated than that, but it gives us a place to start. And that we've shown that there are these persistent effects of early rearing environment, both negative in terms of hypoxia on growth, and positive in terms of warming on stress tolerance. And so what we're just finishing up now and are about to publish is that Annalise's 3rd chapter basically took this information, and we went into the American River, which is just outside of Sacramento, that has these differing warming and hypoxia conditions throughout the season. We put in these tubes basically of embryos, and we looked at sort of hatching success, development, growth in these egg tubes through time to really get a better understanding of what's happening in the field in response to warming and hypoxia that we're measuring at a really fine scale. So with that, I'll thank you for being here. I want to thank our funders at Delta Stewardship Council and NSF, and really thank Annalise for a fantastic thesis, and then all of our collaborators. Thank you.